afternoon, everybody, depending on where you're joining us from. I see we've got some West Coast folks and some East Coast folks. So thank you as always for joining us. My name's Imagine. I'm your host for the TUFC webinar. I am joined here with Kanie Donaldson from UT and also Kevin Bolger from TUFC. And today we have another awesome speaker that I'll be introducing in just a moment. But we're going to go ahead. We're going to go over your CEU information right now, and then I'll give you guys a quick overview of our wonderful speaker today, and we'll get the talk started.
All right, everyone. So if you're here today seeking ISA CEUs, please make sure that you are registered and that you've paid the $10 fee and that your full name is showing to get credit. And if you need to change that, you can go to the participants tab and rename yourself. No payment is required for general attendees or TUFC members. So again, it's free for TUFC members, but if you're seeking those ISA CEUs and you're not a TUFC member, please make sure that you've registered and paid the fee. At the end of the webinar, we will show a QR code where you can also scan it. That just helps us when reporting those CEUs to ISA. Kevin will also drop a link in the chat if you're not able to scan that QR code. Also, during the webinar, if you have any questions, please drop those in the chat and we will get to those at the very end of the webinar. And I think that's it. So Imagine I'll send it back to you.
Lovely. Thank you so much, Katie, as always. Today, I'm really excited, as always. I'm usually very excited to introduce every one of our speakers, but this one I'm especially excited for because I got to see Dr. Georgios Arseniou do this talk or a version of this talk at the ISA Southern Chapters Conference this last year. I actually got to introduce him for his talk and I got to listen to the whole thing. And I thought it was so gosh darn interesting. And so I recommended him to come speak today, and he's been so generous with sharing his time. So today, we have a speaker by the name of Dr. Georgios Arseniou, like I mentioned. He is an assistant professor and extension specialist of urban forestry at Auburn University. He has a PhD from Michigan State and international training in forestry and geoinformatics. His work focuses on using advanced tools like terrestrial laser scanning to better understand urban forests. So I think that's going to be a lot of what we're talking about today. Through his research and extension work, including leading up the Green Up Alabama program, he helps communities build stronger, more sustainable urban forests, and he trains the next generation of green industry professionals every single day. So I will stop talking. Giorgio, if
Uh, can you hear me?
Yes, I can hear you, but I think we lost Imagine, but I think you're good
Okay.
to go ahead and start.
Okay. So, Imagine, thank you so much for the introduction. I also want to say thank you to all of you, Kevin, Katie, Imagine, for inviting me to this webinar today. It's my pleasure to be here. I'm very excited. It's always, my pleasure to talk to practitioners, urban foresters, city foresters, very knowledgeable people, certified arborists, and so forth. So, as Martin mentioned, my name is Georgios Arseniou. I'm an assistant professor and extension specialist of urban forestry in the College of Forestry, Wildlife and Environment at Auburn University. And today, I'm going to talk about advances in urban tree structure measurements and implications for ecosystem services. So, first of all, when we talk about ecosystem services, what do we do really mean? Well, this is also oftentimes referred in the literature as the benefits, or urban forest benefits, right? So we all know that urban trees, urban forests provide very important ecosystem services to our communities, such as cleaning the air that we breathe by uptaking air pollutants with their canopies. Also reducing the heat by casting shade or through cooling the atmosphere through transpiration. This is a very important benefit, especially in warm climates like here in the South, for example, in Alabama or in other parts of the country. Um, Also, water purification is a very important benefit of urban trees. even flooding mitigation, actually. Of course, urban trees cannot prevent floodings, but they can mitigate the severity of floodings. Mental health. I think this is, we're all familiar with this benefit of urban trees and urban forests. Just imagine a day that, has been very stressful for you, and then you decide to take a stroll down the street where there are like beautiful trees. You immediately feel nice. Now imagine if you lived in a place with no trees or very minimal tree cover. That would be not as good. Uh, Also, trees help us to build community. Like, for example, today we are gathered here because we're all genuinely passionate about trees, urban trees. So, this helps us to build a community. And very importantly, resilience. So, urban forests provide a very important ecosystem service, which is uh, uh, creating more resilient environments for people to live. And specifically, when we talk about global warming, urban heat islands, and climate change, we know that urban trees play a very important role because they sequester large amounts of carbon dioxide from the atmosphere, which, as you know, is the main contributor greenhouse gas for global warming and climate change. So my talk today is going to focus a lot on carbon sequestration, urban tree biomass, and how this very important ecosystem service actually relates to tree structure. And then, of course, what type of measurements we need to use in order to quantify tree structure and then carbon sequestration. So that will be the main focus of my talk today. So, as I mentioned, the very important ecosystem services of urban trees relate to their structure and architecture. So, oftentimes we use the term structure or architecture, um, but understanding the structure or the architecture of urban trees is critical to better understand and also quantify the ecosystem services of urban trees, which I presented at the beginning. So, what is actually tree structure or tree architecture? Well, it is the result of the allometric growth of trees. In other words, this means that trees decide how to allocate the available resources in order to grow their branches versus their stems, their roots, and so forth. So this is what we call allometric growth. And the structure, the architecture of trees is the result of this allometric growth. Now, the architecture of trees is based on the interaction of their inherent genetic blueprint and also the environmental factors where they grow. Because as we know, tree structure or tree architecture is very plastic. Trees are very plastic. Their architecture can change depending on the environment where they grow. You may have two trees of same species, but one grows in a forest stand and another tree grows in an open growing environment. They will have very different architecture because of their plasticity. Now, to quantify and characterize tree structure and architecture in traditional forestry and urban forestry, especially when we do urban forest inventories, we use some variables such as tree height, DBH, diameter breast height, crown dimensions, and also biomass. But we also need to keep in mind that, especially for open grown urban trees, we need to consider their branches and the structure of the branches of trees because a large component of the above ground architecture of open grown urban trees is their branches, right? So we cannot omit that very important part of open grown urban trees. And as we know, open grown urban trees have a space filling character, and their branching network actually explains or is an important part of this space filling character of open grown urban trees. So, how do we characterize tree architecture. Let's take a closer look into the architecture of trees and then some key theories that help us basically to characterize tree architecture. So, um, trees basically have a fractal-like architecture. And we can use principles from fractal theory or fractal geometry to quantify tree architecture. Now, what is a fractal? Fractal is basically an object that is self-similar at different scales. So its geometry is repetitive at different scales. Leonardo da Vinci was actually the first one who observed that the total cross-sectional area of the branches of trees is preserved at different scales. And this explains the structural complexity of the branches of trees. Now, here's like a more like kind of like artistic illustration of this concept of the fractal-like architecture of trees, where we see repetitive branching patterns at different scales. All right, so assuming this like fractal-like architecture of urban trees, there are some important theories from biology that help us to understand how trees grow, such as the pipe model theory and the metabolic scaling theory, or WBE model. It's also called West, Brown, and Enquist model. So according to the pipe model theory, we assume that the structure of trees is basically a confluence of unit pipes that connect the roots to the leaves. And these pipes are the structure of these pipes is self-similar because their cross-sectional area is preserved at different scales. Now, according to the metabolic scaling theory, there are also some rules, let's say, that describe fractal-like architecture. So, for example, every parent branch has two daughter branches, and every daughter branch has half the diameter and 1/3 of the length of its parent branch, and so forth. Now, obviously, we know that trees are not like pure fractals, right? Because the branching architecture is not really self-similar across all different scales. And particularly the inherent fractal architecture of trees is disrupted by competition for sunlight. However, open-grown urban trees have a more evident self-similar character compared to forest-grown trees because they face less competition for sunlight or even no competition for sunlight. So, as I said earlier, if we have two trees of same species, one is like growing in a forest stand and the other grows in the open, the tree that grows in the forest stand, due to competition for sunlight, will allocate more resources in height growth in order to reach the upper layers of the canopy because that tree is limited for sunlight. But the open-grown tree is not really limited for sunlight. So it develops a more like squat structure, so short trunks and expanded crowns. So that's why open-grown urban trees have a more evident self-similar or fractal-like architecture. In general, based on previous studies, trees should be better characterized as disrupted fractals because their inherent fractal-like architecture is disrupted by competition for sunlight. All right, so now that we have a better understanding of what is actually tree architecture and what are the key theories from biology, tree biology, that can explain tree architecture, I also want to mention here that one important aspect of tree architecture is biomass. Biomass and then, of course, carbon content, because carbon, as I will mention in a little bit, relates to the tree biomass. So, as we know from the very famous theory of specific relativity of Albert Einstein, the biomass of or the matter, right? The mass is and the energy are actually the two sides of the same coin. So they're interrelated. In other words, this means that if we quantify the urban forest biomass in an urban forest ecosystem, we can better understand the energy of flow within the ecosystem. So that's very important actually from ecological perspective because urban forest ecosystems are complex ecosystems. They're not just landscape trees, but they're like complex ecosystems. So understanding the biomass or quantifying the biomass of urban forests actually helps us to better understand the energy flow within the ecosystem. And now I also want to take a minute here to just clarify that oftentimes we use the terms mass and weight interchangeably, but they're actually different. They're not the same. So mass is a measurement of the amount of matter that something contains, whereas weight is the measurement of the pull of gravity of an object. So this means that the weight of an object can change depending on how far we're from the center of the Earth, but the mass of the same object will always be constant. And of course, they're measured in different units as well, right? Uh, so oftentimes we use these two terms interchangeably, but there is actually a difference between those two terms. Okay, so urban trees, like all plants basically, they photosynthesize in order to produce glucose, which is their food, right? In order to grow and survive. So they absorb carbon dioxide from the atmosphere, and then they build their biomass. And biomass is the amount of biological matter in a tree. Either it is like in the wood structure, in the leaves, in the roots, and so forth. And 50%, about 50% of this like green biomass of trees is moisture. So if we subtract the moisture, then the remaining 50% is actually what we call dry biomass. This is actually what we consider as biomass. After we remove the moisture. And then 50% of this dry biomass is carbon that has been sequestered from the atmosphere. Okay? So in other words, 25% of the total biomass of a tree, including the moisture, is carbon. Now, as I said, carbon is like 50% of the dry biomass or 25% of the total green biomass of a tree. So now you also understand how biomass and carbon basically are related. Now, why is this important actually? What are the implications? So, probably you know already, but an individual tree can store up to, let's say, 0.5 to 3 kilograms of CO2 annually. And of course, this depends on the species, on the size of the tree, on the growing environment, on the age of the tree, and so forth. This is just a rule of thumb here. And of course, this may not seem like a large amount of CO2 at the individual tree scale. However, if we expand the spatial extent, and if we include more trees, actually, this becomes more significant. So, for example, urban forests in the US sequester about 22.8 million tons of atmospheric CO2 annually, based on previous studies. So this is actually very significant ecosystem service of urban forests, as you can understand. And nowadays, cities can also participate in forest carbon markets in order to claim credits for carbon sequestration. For example, the City Forest Credits is a nonprofit that facilitates this process and helps cities and municipalities to go through this through this process for claiming carbon credits. And this is often an overlooked kind of like ecosystem service of urban trees because it's not something tangible. It's not something that you can see. It's not something that you can feel. It's not like shade, for example, that you can feel shade during a hot day in the summer. So oftentimes carbon sequestration is overlooked. However, when we put a dollar amount next to it, it becomes more tangible and people can better understand the importance of carbon sequestration. And then, of course, this gives more incentives to cities and municipalities to plant more trees. Because, for example, between 2009 and 2014, we lost about 36 million trees on an annual basis, which is a huge amount of trees, right? So we want to plant more trees and we want to incentivize cities and communities to plant more trees. So there are important implications for studying this very important ecosystem service of urban trees, which is carbon sequestration. All right, so how can we actually quantify the carbon content of urban trees? Because obviously, a city that wants to claim credits for carbon sequestration, they need to do an inventory. They need to know how much carbon stocks they have in their urban forests. So, how can we do that? There are different ways. The most accurate, but also the most laborious way, it's what they call destructive sampling. So destructive sampling means that we literally harvest trees and we dissect them into, well, first of all, we separate the leaves from the branches and the stems, and then we dissect all these different components into sections. We derive discs and we measure their mass, their grain mass in the field. Then we take some samples back in the lab. We put them in the oven to get rid of the moisture. Then we quantify their dry biomass. And then from that, we know that about 50% is carbon. So this is a very laborious type of work. It takes a lot of time. Several people need to work together to do this type of work. And obviously, it's not logistically feasible in urban settings where we want to plant more trees, not harvest trees for the sake of science, right? So this becomes this method, although it's very accurate, it's not really practical in urban forestry. Now, this is another way that we can actually come up with estimates of biomass and carbon. And it's an indirect method using allometric models or equations, to put it in a simple way. So with these equations, basically, we predict mass or carbon as a function of some easily measured above ground variables of trees, such as dBH, height, and crown dimensions and so forth. So if we measure dBH or height for a given species, then we can plug in this number in our equation and it will give us an estimate of carbon and mass. Now, in order to produce these equations, we still have to do some destructive sampling because we need to actually create the equations, right? So we need to come up with some estimates of the coefficients, as you can see here, for example, the A and B. But just, for you, it's important to know that in order to create those equations, we still need to do some destructive sampling. And based on statistics, the rule of thumb says that we need at least 30 trees per species in order to come up with, in order to have enough statistical power to create an equation for a given species. So this method, although it's not as, it's a little bit better than the previous method in terms of, logistics, but still requires some destructive sampling. So it's not perfect, right? There is challenges. With these allometric models, as I said, we can use some easily measured variables such as dBH or height and so forth, and we can predict carbon and biomass. But there are important challenges because in order to create these equations, we need to sample large amount of trees of different sizes and different species at different spatial scales when we do an urban forest inventory in order to create those equations. So there is a significant time and financial cost associated with this process. And in order to produce these equations, as I said, we still need to do some destructive sampling, which is not feasible or practical always in urban areas. It's very hard actually to convince people to harvest trees to create equations. Uh, so we basically need another way to do that. We need another method, another technique. And this is where actually novel technologies come into play, such as terrestrial laser scanning. Probably you are more familiar with the term LiDAR. It's basically the same thing. This is the terrestrial laser scanning is basically ground-based LiDAR. So what it is actually, it's a laser that is mounted on a tripod. That's why it's called ground-based or terrestrial. And this laser emits millions of laser pulses per second. And each time that the laser pulse hits a target on a tree, let's say on a stem, on a branch, on a leaf, twig, it records a 3D laser point. Um, so we scan our trees from multiple directions, different angles, to have a nice cover of the entire tree structure. So that way we can actually capture tree structure or tree architecture non-destructively, okay, without having to harvest the trees. Now, the data that we collect with this type of technology is called point clouds. Um, Point clouds, they're called point clouds because it's basically like a cloud of laser points, right? And these point clouds consist from millions of laser points. We can either have a point cloud of like a group of trees together, like as you can see on the left side picture here, or we can have like point clouds of individual trees, such as the point cloud of that English oak, open-grown English oak tree on the right side. And as I said, these point clouds, they consist from millions of laser points. So they capture tree structure in much detail. And obviously, now these are like 3D objects, right? 3D representations of trees, but these are 2D images here. But they're actually 3D objects. Now, I also have here an example of the point cloud of the entire forest area, basically, or like an urban forest patch that we laser scanned near Auburn University campus. So this is Forestry. This is where we take our student courses, and it's mostly covered by lowly pine trees. But as you can see, when we scan an area, basically captures everything. These are not like ghosts here, by the way. These are just people that have been captured in the point cloud. But as you can see, we capture the entire scene, like trees, structures of any type, like vehicles, anything. And then, of course, we focus on what is important for us, what trees we want to study, right? So we can segment our point clouds and extract the trees that we're interested. Now, this technology was first uh, used by engineers and surveyors when they want to scan buildings, when they want to build new roads, and so forth. And then in the early 2000s, foresters realized that we can probably use this technology in forests as well and use it to enhance our inventories. And since the early 2000s, we have a tremendous and huge we've seen a huge progress in terms of data captured, data analysis, algorithms, and everything. All right. Now, I want to also talk a little bit about the quality of this data because I don't want to make you feel like this is the perfect technology and the data that we collect is perfect. It's awesome. Yeah, it's awesome, but it's not perfect. So the main problem that we see with this type of data is what we call occlusion. Occlusion means that there are like parts in the point cloud in or parts in the structure of the tree that have not been captured in the point cloud. So basically, it appears as like a gap. And this happens because the laser pulses cannot penetrate the material, cannot go through the wood, cannot go through the leaves. They go through the gaps, which means that if there is like an object in front of the laser scanner that blocks the view of the laser scanner, then what's behind that obstacle basically cannot be scanned, cannot be captured. So then we have occlusion. And here we have an example of two-point clouds of Norway spruce trees, where you can see that on the right side, basically, we're missing almost the entire lower part of the tree. And this is because of occlusion. Now, this is a typical problem, and in order to fix it, we scan our trees from different angles, and then we take all these scans together and we merge them. So eventually we create one single point cloud per tree. This is how we can actually cope with the occlusion problem. And then, of course, we have algorithms that we use to model tree structure, which are smart enough, basically, to work around this issue of the occlusion. So unless you have like a severe occlusion and you're missing entire part of the tree, such as this like point cloud of the Norway spruce here, where we're missing the entire lower part, unless we have a situation like this, in most cases, we're able to reconstruct tree structure when we have occlusion. Because as I said, we have protocols that we can implement in the field to reduce occlusion, and also we have algorithms that can work with this problem. So, in other words, occlusion, there is always some level of occlusion in our point clouds, but what we want to do is to minimize occlusion so that we can get better estimates of tree structure. And occlusion depends on the size of the tree. Larger trees have more branches, more leaves, so more occlusion. Depends on the species. So we typically see more occlusion with like conifers, but again, it's not a huge problem. Like I've scanned longleaf pine trees actually here in Auburn, and we've got some great point clouds that were able to model their structure. Yeah. All right. So once we have laser scanned a tree, the next step is actually to model the structure of the tree. This is what we call quantitative structure model, or QSM. So here, for example, we see on this picture on the left side we have the point cloud of a honey locust tree that was laser scanned in winter time with no leaves, and we artificially colored this point cloud with brown, so it's not the actual color of the tree; it's just an artificial color that we used. But as you can see, we capture a lot of detail actually, even like smaller branches. And then on the right side, we have the model that we created, the quantitative structure model, or the QSM. So how is this model created? Well, we fit cylinders into the point cloud of a tree because cylinders can approximate a tree structure. And these cylinders are basically fitted in a way to mimic tree structure, to follow the topology of tree structure, the actual topology of tree structure. Now we don't do this by hand or we don't do this manually. We have algorithms that do this work for us, right? Um, and because we know the dimensions of these cylinders, we know how large those cylinders are. Eventually, we can quantify tree structure non-destructively. So, for example, the total above ground root volume of a tree will be the sum of the volumes of all the individual cylinders, right? And we get this estimate non-destructively. So just to illustrate how these models work, actually, I'm going to show you a video. So I will stop sharing my presentation and then I will jump to um, YouTube and I will show you a video there basically. So let me share my screen. Yes. Okay. I will enlarge it. And yeah, so this is a video that illustrates the model, the QSM. So, oops. So as you can see, we scan our tree from different angles. This is the laser scanner and the laser beam. We do this from different angles to minimize occlusion. Oh, the video looks a little bit blurry, but uh, hope you can still get the idea here. So this is the point cloud of a tree that consists from millions of laser points. And as you can see, capture a lot of detail. And now we can start fitting those cylinders to approximate tree structure, what they call QSM, right? So we start at the base where we fit our first cylinder. This is what I call subsegment. So we fit our first cylinder and we try to find the best fit. So we minimize the distance of the laser points from the cylinder surface. And once we find the best fit, then we repeat the same process and we basically convert the entire point cloud into a model of cylinders. So we just complete the stem and then we do the same process for the branches. So eventually we have a 3D model of tree structure, which is what I call a quantitative structure model. And we capture a lot of detail, as you can see. So this is what I call quantitative structural modeling, or QSM. And of course, you can search and find lots of information, lots of papers on this methodology. I just want to let you know about it and illustrate the method. So I'm going now back to my presentation. Let me share my screen again. Oops, stop sharing, and let me share my screen again. Okay. All right, so now we're back to my presentation. Uh, so now hopefully you understand better what is actually a quantitative structure model or QSM. I know sometimes it's difficult to wrap your mind around this methodology. My students usually struggle to understand what is the difference between a QSM and the point cloud. Even though I explained to them many times, I always tell them that this will be on their exams and everything, but they still get confused. So I understand it's difficult to wrap your mind around this idea, but hopefully the video that I showed you illustrates the point. All right. So with this technology now, we can actually enhance our inventories. We can simultaneously collect data on several trees of different sizes and different species without having to harvest them. We can easily measure DBH of trees of trees of different sizes and large number of trees simultaneously by measuring basically the diameter of the cylinder that has been fitted, 4.5 feet above the ground. We can also quantify the height of trees by subtracting the lowest laser point from the highest laser point. And there is some uncertainty here sometimes because it depends on how well we have captured the top of the tree. So there's lots of research actually on this topic on height measurements. And then we can also estimate the above ground wood volume of a tree non-destructively if we sum all the volumes of all these cylinders together, right? And then we can also quantify the biomass of trees non-destructively. But this is a two-step process. We cannot directly measure biomass from a point cloud or a QSM. So it's a two-step process. So first of all, we estimate the volume of a tree from the cylinder's volume, which I explained earlier. And then if we multiply the volume that we estimated with the basic density of the wood of a given species, then we can convert volume to biomass. So that's how we can estimate above ground woody biomass of trees non-destructively. And I will show you actually in a little bit some graphs from one of my previous studies where we did this exact thing. Um, and then obviously we don't want to just estimate the biomass of a single tree, but we want to do this for several trees. And sometimes we cannot scan all the trees in the entire city. So then we use some sampling statistics to extrapolate. So what we can do, for example, is we can create zones in our area and scan trees within those zones. And then if you know the area of those zones, of those strips basically, and you know the total area of interest, then you can extrapolate the estimates from those strips to the entire area using these like sampling statistics. In traditional forestry, this is called strip cruising, basically, when we do inventories in forest stands, but the same concept also applies in urban forestry. And then don't forget that we can also estimate carbon through biomass because carbon is about 50% of biomass, right? So you see now how all these pieces are connected together. All right, now I want to show you some results from my previous study where basically we evaluated the accuracy of biomass estimates using this technology because we want to see basically how accurate are the numbers we get, how close to reality they are. So what we did in this study is that we laser scanned um, urban trees and also forest grown trees. And then after we laser scanned them, we harvested them. So we got permission to harvest them. And we quantified their biomass using destructive sampling, which was, if you remember, it was the first method that I mentioned. So that was basically our reference. We had the reference and we're able to compare against the estimates of biomass that we estimated through TLS. And we were able to separate the total woody above ground biomass into main stem biomass and branch biomass. So what we have here now, what we see is we have on the y-axis the TLS-based biomass in kilograms, and then on x-axis we have the reference biomass in kilograms. I know it's in metric system, but we had to use the metric system units here in order to publish the study. So yeah, bear with me. Then the dashed line is the 1 by 1 line, which basically means if like the TLS-based biomass estimates were 100% accurate, then they would exactly fall on this dashed line. And then we have the two regression lines. So with blue, we have TLS-based biomass estimates using density values that we measured in the field when we did the destructive sampling. And then the brown one are TLS-based biomass estimates using published basic density values because there are also publicly available density values for different species. So we wanted to see basically if it makes any difference when we use these two different sources of density. And what we found, as you can see here, is that there was a strong agreement between total above ground biomass from TLS and reference biomass for total AGB, mainstem biomass, and branch biomass, either using reference density values or published basic density values, which is very promising. Now, there was some underestimation, especially for larger trees when we use published basic density values, but this is something we expected. Of course, it's always more accurate when you have measured wood density values, let's say from cores. Okay. And now I want to talk a little bit about some recent research that we've done here at Auburn University that focuses specifically on carbon, urban forest carbon. So this is the work of one of my graduate students who just graduated. And you can see him here in the picture collecting data with his increment borer. So we just submitted these results to journals for publication, and hopefully these two articles will become publicly very soon. So what we did here is that we sampled 90 trees of 3 species Quercus falcata or southern red oak, Quercus laurata or overcap oak, and Taxodium disticum, the bald cypress. So we sampled 30 trees per species in Auburn, and these were like trees both in streets and parks. And we scanned the trees with the leaves and without the leaves in wintertime. But we also derived cores using this increment borer because we want to quantify density, wood density at the tree level, but also because we want to quantify the carbon content of these trees more accurately. We don't want to just assume that it's 50% of the dry biomass. So what we did is that we took those cores and we used another technology. It's called carbon hydrogen analyzer, where basically you combust these like cores and you trace the gases that are released and you trace how much carbon dioxide is released. So then you come up with a more accurate estimate of carbon at the tree level instead of assuming that it's 50% of the biomass, of the dry biomass. So if you know the total above ground biomass of a tree from TLS, and then you know the biomass of a core and the carbon content of a core, then you can extrapolate or you can blow up the numbers from the core level to the entire tree level. All right, so now here we have some results from carbon sequestration in increment cores from the core. So what we found is that there was no difference between the two oak species in carbon content from the cores again. These cores were derived at dBH height, by the way. But the Taxodium disticum had significantly larger amount of carbon, mostly because conifers have more lignin in their wood cells, and we know that lignin relates to more carbon actually, more carbon stored. Now, here we see the differences between street trees and park trees from cores again. So what we found is that for the two oak species, there was more carbon stored in park trees versus street trees, probably because street trees are more stressed due to all the reasons that we know, all the factors that we know, but there was no difference between the carbon stocks of park trees or the carbon content from the cores of park trees and street trees, which is also interesting. Taxodium disticum trees, as we know, bald cypress, they do very well in like swampy areas, wet soil, so very anaerobic soil conditions. So this could be also why there was no difference between park trees and street trees, because these species can do really well in anaerobic conditions, for example, under compacted soils, on streets, and so forth. And now here we have the total above ground carbon content of trees using TLS. And also the carbon hydrogen analyzer, right? So now we're going from the core level to the entire tree, and and we compare park trees versus street trees. So on the y-axis we have the total above ground carbon biomass of a tree on log scale, and then on the x-axis we have the age of trees on log scale. So what we see here is that first of all, the park trees were older than there were more older trees in parks versus streets. But for trees at the same age, park trees accumulated larger amounts of above ground carbon compared to the street trees. Okay, then we also took a look at the allocation of carbon in stems and branches, main stem and branches. And what we found is that when all species are combined, we found that trees allocated more carbon actually into their main stem versus their branches. And that was also true for the Quercus falcata and the Taxodium disticum. For the Quercus laurata, the overcap oak, it was the opposite. We also took a look at the carbon content per branch order, because as you realize from TLS, you can get very detailed information about tree structure, including branches. And as you remember from my first slide, branches are very important in urban tree architecture. So we found that there was more carbon allocated into lower branch orders because the typically larger. And then we also quantified the carbon distribution per branch base diameter. So based on how large its branch was. So we found that there was more carbon allocated to medium-sized branches and large-sized branches. Which also makes sense because these are like, they accumulate more material, so more carbon. And finally, this is our last result here. We wanted to compare the estimates of carbon that we get from TLS to I3 estimates. So as you probably are familiar, I assume you're all familiar with I3. I3 is a publicly available tool for environmental assessment of urban trees through the U.S. Forest Service. So the I3 Echo particularly gives us estimates of carbon stocks of trees at different spatial scales. Now, I3 Echo uses equations to estimate the carbon content of trees, which, as you probably remember, is the second method that I covered in my talk. But these equations originally were developed for forest-grown trees because it's more difficult to harvest trees in urban areas to create the equations. So, because forest-grown trees and open-grown trees are different, there is this assumption that if we just use an equation for forest-grown trees and apply it to open-grown trees, the carbon content of open-grown trees will be inflated, will be overestimated. So that's why I3 Echo multiplies with an adjustment factor. So basically, it estimates the carbon content using forest-grown equations for forest-grown trees, and then it multiplies the estimate by 0.8. So this is what we call CLE, or crown light exposure. So if you use I3 Echo, that 0.8 basically is what I3 Echo refers to as crown light exposure parameter. And you can have it on or off when you use I3 Echo. So what we found here is that there was always a strong agreement between I3-based carbon estimates and TLS-based carbon estimates, especially when this CLE parameter was not applied. So basically, we found very strong agreement between the two methods, I3 Echo and TLS, when the crown light exposure was not applied. And again, the crown light exposure is a parameter within the I3 Echo. Okay, so that's something interesting that tells us something about I3 Echo and also TLS. Yeah. And finally, here are some resources if you're interested to find more information about this technology. I have some research studies here, but also some extension publications that you can use and find more information. So with that, I want to thank U.S. Forest Service for funding my work, and also the city of Auburn and Auburn University for allowing this research actually to be done on, in our city and on our campus. So with that, thank you very much for your attention. And if you have any questions, feel free to ask me or reach out to my email address. So thank you very much.
Thank you so much, Giorgio. I got to hear a version of this at the ISA Southern Chapter Conference this last year. And I thought it was a very interesting subject that isn't talked about enough, at least in our spaces. And hearing it again, I'm pulling out all these new pieces of information that I didn't before. So just really interesting. I really appreciate you, sharing that with us. That was a very good talk.
Thank you.
Dense, a lot of good stuff though. Love that. And so with that, Katie, I'm going to push it over to you. I know you've got to get through the CEU information. And I did see that we had a question, at least one in the chat. If you have any other questions, throw them in now. And I know we've only got a few minutes before the end. So go ahead, Katie, take it away.
Thank you. And real quick, if you don't mind, I'm actually going to take over sharing my screen.
Oh, sure. Yeah, I'll share mine.
That way I can pop up the QR code for everybody. Okay. And then I will ask questions. So first question we got, how expensive is the terrestrial laser scanning? Is the technique affordable or feasible for non-institutional users?
And that's a good question. So The laser scanners, the price of the laser scanners can vary. Typically, they're within the range of like 50,000. Uh, but drone-based LiDAR actually can give you also very good data in much lower cost. So I would suggest drone-based LiDAR for commercial purposes. I know there are like trickier businesses that use actually drones a lot, either collecting images or LiDAR data, and it's cost efficient, very cost efficient, and gives you good data.
Okay, thank you.
Can I,
Um,
Katie,
oh,
really quick? I'm just curious to follow up on that. So, in your opinion, for people that are trying to kind of get this data in their urban settings, do you still feel that like iTree for like the common lay like most users would want to use that system just kind of with those attributes you mentioned turned off?
Yes, that's also a good question. Yes, absolutely. iTree gives very good estimates of carbon estimate, and as we can see, at least based on what we found here, there was a better agreement when the CLE parameter was turned off. So yes, of course we need more studies to better support this finding, but based on our study, which we're going to publish very soon in a journal, when the CLE parameter is not used, we get better estimates of carbon.
Okay, thank you.
Because also this like 0.8 adjustment factor, there is lots of studies out there basically that question the accuracy of this factor. Yes.
okay.
And I see another question here also, like how many minutes per tree it takes to scan. So it takes us about like 20 minutes per tree. 20 or it also depends on the size, so it can also go up to like half hour, but that's usually how long it takes us per tree. Now, this is if we want to scan individual trees. As I said, we can also scan several trees together, right? So we can expedite the process. We just need to come up with a different pattern and mount the laser scan at different locations so that we have a better representation of the entire forest, of the entire group of trees. You probably remember in one of my slides, I showed you that we can have point clouds of trees individually, or we can scan the entire group of trees. Um, so I see maybe one more question. So it says you can actually do a comparison of the benefits between trees using MyTree, which uses same method as iTree Echo, but is a little easier to use. Yes. Yes, actually, yes. I use MyTree actually a lot in my extension programming when I teach students about urban tree benefits. So yeah, that's a good point. I3 Echo is more like for professional purposes, for like urban forest benefits assessment at different spatio-temporal scales. So it's a little bit more sophisticated.
And then Giorgio, I do think there was one other question, and it
Mhm.
was asking, what about the carbon stored in the roots? So are you guys going to be looking
Okay.
along that?
That's a good question too. So, with this technology, we cannot actually penetrate the ground to scan the roots below the ground. So, in order to do that, you need different technology, like a radar technology, that can penetrate the ground. So, yeah, with LiDAR, we cannot penetrate with this type of LiDAR, we cannot penetrate the ground.
Is that something that you might be researching in the future? Just curious.
Oh, yeah, actually, yes. This is something I've been thinking a lot, and I'm trying to find collaborators. So, if there is anyone basically that would like to collaborate on this idea, I would be very excited to work because we are missing a lot of information below the ground. Very quickly, I'll give you an example. I was talking to another researcher. There was a site in Florida where some trees were basically they were fell down due to during a storm, a severe storm, and the roots were exposed. So it was the only time basically that we could see at least a portion of the root system above the ground. And we want to scan those exposed root systems. Of course, it's not the entire root system, just a portion of it, but it still gives you some idea. So if there's anyone who is interested to do this type of work, I'm very excited to talk more about it.
Well, I think those are all the questions we have. Let me know if I missed any. And just as a reminder to anyone who is still on this webinar, you can scan the QR code or click the link that Kevin dropped in the chat to verify your CEU attendance. But I believe that is it.
Thank
Awesome.
you.
Well, thank you guys so much for joining us for another TUFC webinar. Thank you, Dr. Giorgio Arseniou, for your time. We have his contact information, and we'll send out the video not too long from now. So thank you guys so much for joining us, and we'll see you
Thank
in.
you all so much. I really enjoyed it. Thank you.
Yeah, thanks for coming.
Thank you. Have
Bye.
a nice day. Bye.